Hello and welcome to this episode of BIOS Frontier Science. Today, I'm incredibly excited to welcome Gordana Vunjak-Novakovic, University Professor at Columbia University. To help kick things off, Gordana, could you share a brief introduction with us?
Sure. So I trained in chemical engineering at the University of Belgrade in Serbia, just like my father, because I really loved the breadth and the creativity of his professional work. All the way through my PhD, which I obtained between Belgrade and Germany, I went through a rigorous engineering curriculum with a lot of math and not a single class of biology, which was my other love. At that time, I also learned about the importance of inventions, and I filed my first patents. I was offered a faculty position at the home department right after graduating, started a family at the same time, so life was good. But something was missing, and I just could not find passion in pursuing classical chemical engineering. I kept thinking if there is a way to put engineering at the surface of biology or medicine, and these disciplines were not really connected, at least not obviously at that time. My department was nice enough to let me apply for Fulbright Fellowship, which I spent at MIT in search of my future area of work. By serendipity, I met Bob Langer. At the time, he was just starting to launch the field of tissue engineering, and I realized my search is over. I was really fascinated, and I still am, by the idea that we can grow living tissues in laboratory. I also, by serendipity, realized that what I studied for my PhD, although it was very classical chemical engineering, provided a foundation for designing bioreactors, meaning that we can build the homes for our growing tissues in laboratory and have both control and insight about how they do. So it's really looking for opportunity that actually brought me into tissue engineering, and I'm very grateful to Bob, who opened this door to me and to many others in our field.
Professor Langer is a great friend of BIOS, and it's clear that he's had a broad impact. So thank you so much for sharing your story. It's also, as someone who was a chemical engineer turned bioengineer myself, it's only because of pioneers like yourself that was a career path so readily available for me. So thank you as well. And before diving deeper into maybe specific projects and research, one question we love to ask our guests: throughout your career, do you have a North Star, the common thread, if you will, that's been tying your work together?
I think we all do. And I think that my North Star is the passion to connect engineering with biology and medicine and to a continuum. And so that we can accomplish something that could not be done within either area alone. This idea of using engineering to enable biology for eventual benefits in medicine is really very unique. I believe that some of the most exciting science really happens at the interfaces of disciplines rather than within any specific area. And when you look into the progress that has been achieved over the last few decades, it really resulted to a large extent from the convergence of disciplines. And when you go out of your zone of comfort and talk to people that have very, very different background and knowledge and expertise than you do, you learn very directly what are some of the scientific and some of the medical challenges of our time. And working and talking with them, you come up with some fresh ideas and fresh concepts on how to address some of these challenges. Also, at the interfaces of disciplines, I think there is a much greater chance that you may have an accidental finding, and that because things are less predictable, and it's a kind of easier to go off tangent in a completely new direction. And the last thing I would like to say is that's related to following this North Star is that the young talent that we work with, our students, our postdocs, our clinical fellows, are really the key ingredient of this entire process. So thinking about connecting medicine and engineering, this can be done in an infinite number of ways, and it's very difficult to decide what to do. And this is actually where our choices can be inspired by the actual needs of the medicine or society. And this is where you will go again back to the people that actually can articulate these needs, and they can direct you into where are the most critical problems, and also what may be the ways to address these problems so that you can like most directly plug into the normal workflow in clinic and really the needle make some kind of difference.
Cool. I love the way you highlighted people there, and on top of that focused on translation and real world applicability, as any good engineer would. And so to start talking, I think, about some of those interfaces, let's turn over to your work in tissue engineering and regenerative medicine. And so your lab focuses on tissue engineering and regenerative medicine with applications in the cardiac, lung, and skeletal spaces, in addition to the design and development of organ-on-a-chip models for biological and medical research. Would you mind giving us just a quick overview of your tissue engineering and organ work?
Sure. So people often ask, how can we work in so many different areas? And the answer is actually very simple. The fundamental principles are really the same for all these tissue systems, for everything we do. To put it into very simple words, our job is to provide the cells with an instructive environment for performing their normal biological functions. So we are really enabling the cells. And we do that by using bioengineering tools. So we are not really engineering tissues. We are enabling the actual tissue engineers, which are the cells, to do this for us. And the two tools that we are using are the scaffolds, or biomaterials, and the bioreactors. So the scaffold is always designed for a given tissue to mimic its native matrix. So it's different for muscle, from bone, from brain. It's very tissue specific. So such a scaffold will provide both the structural and also informational template for the cells to build a specific tissue type. We also use bioreactors, and bioreactor is a kind of substitute body because it is providing homeostasis, so the normal environment for the cells, exchange of nutrients, most critically oxygen, and also the provision of regulatory factors, molecular and physical. So this is what's the commonality. So our designs of different tissue systems are obviously different because they are informed by biology. This is another very important principle. So it's really biological foundation and engineering designs. And I must say that our challenges are also in the area of biology. If you know what are the conditions that you need to provide, you always find a way to do so. So engineering is a kind of easy. Biology is what is really difficult in this combo. So we are in these different organ systems, we are very interested in also recapitulating different situations. So we study development, we study injury, we study disease conditions, we study regeneration. And we also work on multiple scales, as you mentioned, because we work with millimeter-sized tissues in organs on a chip to provide biological models for biological research, all the way to the whole organs to work on regenerative engineering. This is where close collaboration with experts from different areas, from cell biology to immunology to oncology, surgery, is really critical to actually bring these specifics of not only each organ system, because the liver is different from heart, it's also to bring in the detail of the diseases and like what are the approaches that we should be taking to recapitulate specific disease condition and to study it and eventually to come up with some new therapeutic options that we can test.
That all makes a lot of sense. And that point you touched on at the end, bringing individuals who are experts in so many different disciplines together to turn this into a more applied and translational approach. As a pioneer who's been really creating and driving this field, can you share more about maybe some of those key interdisciplinary aspects and what enables you to bring them together?
Yeah, so what is our goal and really our passion is to build the tissue models for biological and medical research that can recapitulate some of the human conditions, some of the conditions that you see in patients in either health or disease. Mm-hmm. And it is, on one hand, it's really basic science research, it's research. On the other hand, this is this innovation implementation cycle that we are trying to drive because we are really, really aiming to eventually translate discovery into benefits for the patients or for the society. So with this in mind, when we look back, we see that the field of biomedical engineering and tissue engineering has followed for several decades this bench to bedside paradigm. So this was the guiding principle. So you would have an interesting discovery in laboratory, or you would develop a technology that is potentially impactful. And then you look how you can find application in medicine or commercial sector. Mm-hmm. So I think that this model and experience shows that this model works really well for studies that are done within more or less in isolation, within one specific area, which can be engineering, can be something else. Yeah. And interactions between engineers and, say, clinicians are starting at a time when research has been more or less completed. Looking forward, I think that the increasingly interdisciplinary science works differently, and it is resulting in changes in this bench-to-bedside paradigm. Looking into the ongoing projects and research projects, what we see is that our work is very often commissioned. It starts from the bedside. It starts from learning from the clinicians about a specific clinical problem. So they also can inform us about the nature of solution that would be valuable to them, because we all know clinicians will not change everything they do in order to accommodate our wonderful technology. So we need to be the ones fitting into their workflow. Mm-hmm. And so you really start from the bedside, you go back to lab, to the proverbial bench to find the solution, and you return to the bedside for implementation. And very importantly, this can only be done as a collaborative effort. So tissue engineering is a team effort, team job. I mean, without it, you can really not make much progress. So the selection of the research project and the nature of research that we do and many others has changed quite a lot, and it's really inspired to a great extent by the needs, by the gaps, by the challenges of our time. So I can give you two examples about how these challenges are really motivating and informing our work. Our lung research has started about 10 years ago with a group of Columbia lung transplant surgeons approaching us about the lack of sufficient numbers of donor lungs for patients that suffer from end-stage lung disease and don't really have any other option but to receive the new lung. This crisis is further augmented by the fact that the majority of the lungs that they receive in hospital is rejected as unacceptable, either because they had some injury during harvest, or there is a disease that was not originally discovered, or simply because they traveled across the country and lungs don't travel very well. They really suffer from hypoxia. So the very small, insufficient number of lungs is very substantially decreased at the site of in the transplant centers by these rejections. So our first reaction was the sort of to push back. We don't work on lung, it's too complex. I mean, everyone knows that lung is one of the most complex organs in the body. So you have more than 50 types of cells, which is incredible. And there is this hierarchical structure of both the vasculature and the airway. And at their interface is the gas exchange surface. This is what the lungs do. I mean, breathing is really, really high on our priority list. So this happens through the surface area that is about a half of the size of a tennis court. So this is incredible. We cannot make a lung, not yet at least. So we started to think if we can lower ambition and recover those lungs that were rejected because they're not totally dysfunctional, they're just not functional enough. And actually this resulted in the development of a new technology that is able to recover many of these rejected lungs to the point that they meet a standard for transplantation. More recently, we also started to work in another direction using this technology, which is to use the whole lungs that we are supporting ex vivo to model different kinds of diseases, for example, fibrosis, either genetic diseases, or we also worked on the conditions that are similar to those. It's acute respiratory distress that is associated with COVID, but also associated with some situations, say someone is breathing in bad air, such as firefighter. So this was one example. The other very different example is that there is a clinical need for tissue engineered reconstructions of large and complex defects in skeletal tissues involving, say, bone and cartilage. And especially almost all our skeletal tissues are sort of anatomically complex, which is even more true in the head and face than in other parts of the body. To this challenge, founded in a completely different way, we can grow bone. We have grown human bones for a very long time. So the problem was only to do it so that the grafts are anatomically correct and that they are customized to the patient. So you can make grafts that are matching the patient's biology and the patient's anatomy. And actually this technology excelled pretty fast, and it is now in clinical trials through one of our startups that we launched from the lab. So two different solutions, but the common point is that both of them came from a clinical need, not from our specific. We also do a lot of projects that come from our scientific interests, but we are really trying to match the two, you know, and to also work on something that may be clinically beneficial.
And as you highlight, by starting from that bench-side approach and focusing on the point of collaboration, you're able to lead to more actionable outcomes for patients more quickly. That sounds like, and just those two examples you gave are phenomenal. If anything, I'd love to dive deeper into both, but maybe we should pause on that for a moment and just dive a little bit more into the foundational technologies that you were describing earlier with scaffold design and stem cell incorporation being brought together through, shall we call them more targeted bioreactors that are a large part of this tissue engineering research. And so can you share a little bit more about this intersection of biomaterial scaffolds, cell seeding, bioreactors, and how bringing these technologies together really advances the projects you're talking about?
Yeah, so I think I already mentioned that the ultimate and only tissue engineering is the living cell.
Yeah.
It is very important to understand that the cells are responding to the entire context of their environment.
Mm-hmm.
So responding means that they can perform and will perform differently under different conditions, either in the body or outside the body in the laboratory. So we are really using the engineered tools that you're mentioning, the scaffolding materials and the bioreactors, to tell the cell what to do. And if we are clever enough and precise enough to convey this information, the cell is clever enough to use it and to go in the right direction. So some of the engineering requirements that we really need to meet when we talk about these tools are, for example, for scaffolds, in principle, they need to resemble the native tissue matrix in all possible ways, as much as it's doable. So they need to have the right composition, they need to have the right structure, the right mechanics, sometimes the right conductivity if you're talking about the muscle or a nerve tissue.
Mm-hmm.
If you're studying the conditions of disease, this will also affect the matrix and needs to be reflected in different properties of the scaffold. Like when we are modeling fibrosis in the heart or lung, or joints, then you have the stiffening. And you would be very interested in incorporating the materials that have this ability to stiffen during the process, either on demand or in an ideal world in response to the cellular signals. The scaffold also needs to be biodegradable, because we like to see it all gone at the time when the tissue formation is completed. And then finally, something that is just about emerging in the field, David Kaplan is one of the people that is pursuing this very actively, scaffolds that have conversation with their environment, with the cells. So scaffolds that are able to sense and to respond to the cellular signals. So these are some of the requirements that we are very interested in meeting. Bioreactors, just very briefly, are also designed to mimic the biological features that we want to study. So they provide a specific tissue-specific or organ-specific environment, which means, again, providing the composition and the regulatory factors that this tissue has in its native environment. The bioreactors also keep the tissues oxygenated or not oxygenated. Sometimes, for example, in studies of cancer, hypoxia is something that you want to establish. And they provide controllable exchange of nutrients and metabolites, because we don't have during our tissue growth cycle, you don't have these other organs in the body, say liver providing metabolism or kidney providing clearance. Bioreactor needs to do it for you. And then finally, from the engineering point of view, it allows you to see things as they happen. So they need to be compatible with imaging. And also we put great value on the online measurements of tissue functionality. So you can, for example, introduce perturbance into the system and see how the tissue responds to do this kind of dynamic studies without touching the tissue. So you just study the same tissue over and over a long period of time. So this is like a nutshell, but these two components together are need to provide to the cells in order to grow and maintain any particular tissue.
That makes a lot of sense. And especially as we're talking about reconstituting the tissue environment, I would assume that things like mechanical forces can be important to consider, especially, for example, when thinking about musculoskeletal tissue regeneration. So it's also, I would assume, building reactors, when we're talking about mimicking the environment, we're not just talking about the metabolic conditions, but also the structural conditions, the physical conditions that sort of prompt cells down targeted differentiation pathways at the right times as well.
I mean, this is really a great comment, and this mechanical, or if you want, physical environment, because it also involves some of the electrical signals, critically important for the formation of the tissue. For example, when we grow a heart or skeletal muscle, we subject this muscle to electrical field signals that are very similar to the signals that the heart cells and muscle cells feel in the body. And this is electromechanical conditioning because, for example, when you expose the heart muscle to electrical field stimulation, the membranes are depolarizing all at the same time, and all cells twitch at the same time. And this is the principle of how our heart beats and pumps blood. Heart is a really great example because this is the first functioning organ in our body. It starts beating like 3 weeks into gestation. You can hardly see the heart, and it's already beating. So you can imagine that providing the developing heart muscle in culture with the same signals that are guiding its development, in its prenatal development is critically important. For cartilage or bone or other skeletal tissue, mechanical stimulation is critical. For tissues like bone, you have another component, which is perfusion. So bone is also subjected to this hydrodynamic shear that is vital for its functionality. And it has to be the right level of the shear and the right dynamics. So it's like the whole field of this mechanobiology of the cells and tissues that came into the picture decades ago and has been incorporated, into our tissue engineering designs.
And as you said, you have to start with the biological understanding, and then you can build the engineering system to support and ideally mimic it for your idealized outcomes.
Absolutely. You do need to respect biology because cells have this biological memory, they have their repertoire of things that can do. So you need to accommodate biology by engineering then the other way around. So, I mean, we can do it. You can do it the other way around, but then the results will be irrelevant and really non-physiological.
I'm going to take this into projection in the future just for a second and ask you a question. So as we're thinking about this, at the moment, the goal is to replicate and mimic what already exists. I'm curious, do you think there might be a future state where we are able to optimize past what developmental biology has led us to today, and instead say, if we tweak the environment, we can create a more robust bone structure, or a more elastic and yet strong musculoskeletal structure, or a heart muscle that's more resistant to fibrotic damage, or something. And I realize this is projecting far out into the future. I'm just curious, as someone leading the field, your thoughts.
Yeah, it's another really nice comment. So I think there are two questions in what you say. One is, can we and do we need to make something that's better than native, like a super bone or super muscle? This is something we need to think about. Like, what would be the purpose? What would be the application where such tissue that is very special has like a superior properties compared to the native tissue could be applied. The other question is, can we do things faster? Because the timing is really very important for regenerative medicine, for diagnosing disease. In other words, can you somehow accelerate normal processes so that you get to your final point sooner? And we know that this is possible because partially by thinking, partially by serendipity, we invented a protocol for engineering and maturing the human heart muscle that actually achieves the some of the adult-like properties much, much sooner than the body. I mean, we developed 9 months before we are born. And then in culture, by exposing this heart muscle to like supraphysiological electromechanical signals, we accomplished this goal over just 4 weeks. And our understanding of this, and actually these were also some comments of this paper, they articulated the idea that this is like training for Olympic Games. You are challenging your body to the extreme, but not beyond the extreme. So it has to be a sort of tempered and timed. And then this muscle that we cultured in its fight for survival was getting stronger much faster than it normally would. So this was like a really, really interesting finding. And now we are looking at these culture systems can provide this acceleration maybe in some other areas. Like one question is, one of the conditions that we are studying very actively is the cancer metastasis, which takes very, very long time in patient to develop. So if you're going to predict if the patient has propensity to develop metastasis, is there any way that we can sort of change the conditions of culture? So if the cells really have inclination to populate distant organs and for metastasis, so that they do it sooner. And obviously this would be incredibly helpful for guiding, you know, the management of any specific patient. So if you know that the patient is more likely than average to develop metastasis, and if you can predict if this happens, which organ would be affected, then obviously clinician can focus on the, you know, on monitoring the parameters that will be most informative for the clinical management of the patient. So there are many, many questions, but the good news is that we are building models. So if you have models, you learn from them and you do better. And then you go back and you do even better and even better. And I think this collaboration and exchange of experiences, information, you know, between different labs is really helping tremendously to make further progress in this incredibly complex area. Again, biology is very, very complex, even under very normal conditions, not to mention the conditions of disease.
Well, I think that's the perfect transition, because I'd love to dive deeper into your work with modeling, especially through organ-on-a-chip technologies. And so as we think about your work with biomaterials and the cell interactions you were even just describing through practical examples to engineer multi-organ chips, you've been recognized with a recent paper also in Nature Biomedical Engineering. Congrats. And so we'd love to hear if you could expand on some of those multi-organ chip platforms and their impact on modeling diseases by efficiently elucidating aspects like drug efficacy, safety, and pharmacological profiles.
Yeah, so organs on a chip really emerged about 10 years ago, more or less. The idea was around in the field, but actually the real effort escalated about 10 years ago. And this happened in response to another clinical need, and this is to make more predictive models of human physiology and disease by combining advances in stem cell sciences and tissue engineering. So this is really the charge that was articulated by NIH, and with NIH support, initially about a dozen laboratories across the country started to develop different kinds of either single tissue or multi-tissue models. So over these 10 years, we have witnessed very significant progress, both in increasing the fidelity of these models, making these tissues more and more like structurally and also functionally to their native counterparts, and also progress in their utility, so they're used by pharmaceutical industry. You could see that the big pharma started to adopt this model. At the same time, there is a number of startup companies that actually they're launched over the last maybe 5 years or so that are commercializing some of these models. So comparing organs on a chip to regenerative medicine, both of them are based on tissue engineering, you see a big difference. We perceive today the organs on a chip as a fast-track application of tissue engineering. And I think this is at least for two reasons. One is that the regulatory procedure is much simpler and faster. Nothing is being implanted into the patient. Everything happens outside the body. And this is also related to the very strong interest of pharma. And the other reason is because the principles are the same, organs on a chip really capitalized on advances of tissue engineering that were motivated by regenerative medicine. So you make grafts, you engineer grafts that are clinically sized, but then the knowledge gained during this process can be translated down to these millimeter-sized tissues, and so the progress was really much faster. So one clarification maybe to bring up, although, I mean, people may know, but just to make clear, these are not really microorgans. We are not talking about the little heart and the little liver and the little brain. These are just tissues that are representative of organs and designed to recapitulate some of the organ level functions. So the liver module is metabolizing. This is what we really want from liver. If you connect heart with a skeletal muscle with a motor neuron, then you get functional neuromuscular junction, and you can model diseases such as ALS or other dysfunction of the. You can model contractility of the heart, etc., etc. So the most sophisticated of these organs on a chip are also those that are connecting multiple tissues by vascular perfusion, so that you go to the next level, which is to model physiology of. You make a physiological unit, so you can study a specific question which involves interaction between different organ systems. So you can, for example, to model inflammation, you would need more than one organ system. To model cancer metastasis, you would need more than one organ system. If you study, for example, effectiveness and toxicity of an anti-cancer drug, you need at the minimum the tumor to study effectiveness, liver and heart to study kidney, to study toxicity, and you need vasculature to make them communicate with each other and carry, the cancer and immune cells. So these organs on a chip, to have utility, they need to meet lots of requirements. So there is a wish list that we are really trying to follow. One is they need to be modular. They need to be a sort of plug and play so that you can configure the physiological units of interest. And you can also, by including different organs, you can also, if you study drugs, you can study different routes of administration, like by injection, by inhalation, by exposure, to skin. Patient specificity is not universally important, but it is it is required for certain diseases that are known to have patient specificity. So myocardial infarction, myocarditis resulting from, say, autoimmune diseases or cancer are example, or any immune disease are example of diseases that have great patient specificity. Moreover, The field is now looking more closely, finally looking more closely into the diversity of patient population. We have the tools now. So we know that the sex, the age, the race, the history of disease, all this is very important. And all this can be our experimental variables at this time, because we did develop systems that can be made patient-specific. Another requirement is that each of these tissues in single or multi tissue platforms need to have the right phenotype, which means it's expressing the features of its adult counterpart. And we need to maintain this phenotype for a long period of time. Long is weeks to months. So this is what we need to study chronic conditions. At the same time, in most cases, you also need these different tissues to communicate to each other. And then finally, as for bioreactors in regenerative medicine, bioreactor also has a function of providing you insight into the tissue, so you can measure it, you can see it, we can track the cells, see the cell migration. So there is this technical requirement, technical component of the bioreactor. So I think these are probably the key requirements that we try to provide.
It sounds so easy when you describe all of the elements and components that are needed, from the spatial temporal considerations to the drill down into matching organ systems and recapitulating them in their, if you want to call it, essence in these microsystems. And it sounds like microfluidics is one of the primary base tools or tool systems that enables organ ownership structures to be developed in the first place. So as we think about advancing these technologies and bringing them forward, and we try to create multi-organ chips, as you described, that are more representative of the human body, so that way they can better recapitulate models of disease. What are, besides the biological understanding, which is clear, are there very strong challenges that need to be overcome? Maybe from a maybe from a translation perspective and the selection of these microtissues or the adaptation of their environments or monitoring. I suspect there are challenges across the board, but forgive me, I'm answering on your behalf, please.
Yeah, so I can mention a few. So one fundamental challenge, again, we go back to biology, how to recapitulate biology. One fundamental challenge was how to reconcile the two requirements that I mentioned. One is to maintain the individual tissue phenotypes over a long period of time, and the other is to allow them to communicate to each other. So this is something that, I mean, the person who really figured it out, and she's the first author of this Nature BME paper that you mentioned, Casey Ronaldson-Bouchard, we talked about it and we came up to this plan, like, why don't we just do what the body does? Each of our organs is happily sitting in its own compartment, which has a boundary, which is selectively permeable, and they do communicate with each other. How do they do that? By vascular flow, by circulation. And between the organ space and the vascular flow, there is endothelium, there is vascular lining, which is selectively permeable. Very interestingly, this vascular lining can sometimes pass, say, immune cells in response to the injury signals, while it stays closed for much smaller entities, such as some molecules. And this is what we have done. We just placed an endothelium between each compartment and vascular flow, and there is a flow, there are organ compartments, and they communicate with each other by secreted factors, by extracellular vesicles, and sometimes you also see the cells crossing this barrier in response to the trophic signals. So this was one challenge, and I think this is something that I really find fascinating, that you can recapitulate some of these fundamental features of biology. The other challenge is really logistic or technical. for a further impact, we really need to disseminate these organs on a chip. This is a real prerequisite for their broad use. They need to be used by many, many labs. They should be as easy to use as, say, a well plate. The chip that we currently have, this multi-organ chip, it has been designed for academic research. It's an artisan device. It requires training. You cannot just push button and use it. And now what we are doing is that we are turning this chip into autonomous push button device that is compatible with plate readers, with imaging devices, with robotics for pharmaceutical use. So it has to be very easy to use while maintaining this sophistication of being instrumented, you know, and have a high capability. So this is really like a big, important a task for many of us, not unique to our lab, I think for the whole field, just to exchange the systems more directly. The other challenge is, I believe, further and rigorous validation of this organs on a chip against clinical data. They're very elegant. They show us in research like many features of the disease, but just like validating them side by side against available clinical data for specific conditions, diseases, tissue systems, and whenever appropriate also against animal models is something that will really increase their acceptance and help build confidence, you know, by users to actually extend their application. And the final one more challenge that I would like to mention is really data synthesis, data interpretation. We are generating increasingly large volumes of data, and they're very different from each other. So how do you interpret together, say, single cell sequencing data and functional data and compositional data? So we are reaching out now. The data interpretation goes beyond statistics, which is still like an obligatory section in every paper that we write. But we do reach out to machine learning, to artificial intelligence, to like higher levels of interpretation of this spatially, temporally non-uniform data. I mean, there is much more, but I think these 4 challenges, one of which is more like on the biological conceptual level, and 3 are more like logistic and related to collaborative efforts, are probably illustrative of where the field is at this time and where it may be going.
As well as the complexity and the potential of the field as well. And I'd be curious, this is again thinking out into the future, but as we consider the development of more and more high throughput and rapidly designed organoid models, do you think there might come a point where we can link tissue-specific and patient-specific organoids with these multi-organ chip systems, to that way not only represent the organs, the systems of the body, but also patient-specific organs as well?
This is already happening in the research space. I mean, I just was yesterday at the conference, it was Nature Cancer meeting, and this is exactly what we talked about, the patient specificity. There are logistic challenges also to this, but I think the importance of doing studies in a patient-specific manner, studying one patient at a time, what we call N=1 study, are so important that everyone is super motivated at, you know, like overcoming these challenges by many, you know, in many different ways. So the methodologies are changing, the approaches are changing. I think we are really going there because it is really very important.
You know, makes a lot of sense, going back to what you were saying about starting at the bedside and then bringing it back to the bench. Well, I know I could keep asking questions on this forever, but to keep the conversation moving through your work, Your lab is also exploring and furthering the future of tissue and organ repair and replacement, including aspects of bone, lung, heart, and cartilage. And we've touched on this briefly, but we'd love to hear more about your work here and its potential for translation into the clinic, and to really understand what role stem cells play in this research.
Okay, so our lab is very translational. So we all believe the translation of science into clinical and commercial applications is as important as science itself. So it really goes hand in hand. And the way we understand translation is actually to follow these two paths. We are translating tissue engineering into regenerative medicine by engineering pieces of tissues, or sometimes re-engineering the whole organ, such as lung, for regenerative medicine application. But on the other hand, there is a lot that you can do using organs on a chip, and we are starting to study injury and regeneration and therapeutic modalities in the context of wound healing in organs on a chip. So what there can happen is you may discover, say, the regulator of certain behavior or an agent that may be helpful, and then you are translating it through a completely different modality to the patients. So I think both of these paths are really important. And as I mentioned, they are both based on same fundamental principles, with the organs on a chip giving you convenience of working on a very small scale with very large number of samples without any regulatory constraints, and finding out useful information that can be translated into patients.
Well, as we think more about that translation, at this point you founded 4 biotechnology companies, including Epibone, Terra, Xilex, and Implicate. And just to highlight one, Terra Biosystems provides, to go back to what you were saying earlier, predictive in vitro human cardiac tissue models for use in drug discovery, safety assessments, and translational medicine. I'd love to hear more about the innovations behind Terra and this intersection, as you were describing it, of biology and data to really generate that insight for translational medicine.
Okay, it's actually a very nice story. It took us about 15 years. So it started in 1999 in Boston. I was still in Boston at that time, and I started developing the engineered heart tissue with my student at that time, graduate student, Milica Radicic, who is today really a big name in cardiac tissue engineering. She's a professor at the University of Toronto. So I started working with animal cells. So this was the time before human stem cells. These are like old time. And moved gradually to human stem cells, learning how to build the muscle. And the principle that we invented in this study is this principle of electrical conditioning of the tissue to make it more functional. So in 2005, after PhD and postdoc, Milica moved north to Toronto. I moved south to New York, and we never stopped working together. We just continued working together, and we ended up developing this heart on a chip system that was very suitable for different types of physiological studies and for testing of drugs. So in 2014, with a lot of support from different sides, we launched the company together with students from our lab. So there are many co-founders of Terra. And because Milica and I are compatriots, we named the company after a mountain in our native Serbia. This is the most beautiful mountain that we both love. As for all our other startups, some of the students and postdocs who developed technology moved into the company. So I mentioned this because I think that this is incredibly important, this continuity, intellectual ownership, and dedication for advancing technology, and because this is your technology that you are developing. You are not hired from outside, to work on someone else's idea. And so Tara, as you may know, was actually it was acquired recently by Valo to be really a critical component in this like vertically integrated platform with cardiac tissue for drug development. And Tara is also an example of a company where we very, very quickly started to reach out to the advanced methods for data analysis that I just mentioned, which are based on machine learning and artificial intelligence. And it was out of need because the diversity of data and the volume of data just required like a very clever, effective approach that goes beyond the, you know, what you classically do.
That all makes a lot of sense, and thank you for sharing more about that story. And to build on some of that success, your research success in tissue engineering and regenerative medicine has led to many awards, including some firsts. The first woman faculty at Columbia to be inducted into the National Academy of Engineering, the first engineer at Columbia to be inducted into the National Academy of Medicine, and the first engineer at Columbia to receive the highest distinction of the university. Congratulations. I'd love to understand, as someone who's pioneered so many paths yourself, what your thoughts are on what we can do to further support and mentor the next generation of scientists, and especially female scientists.
Sure. So thank you for what you're saying. I mean, these awards are very nice, and I really see them as honors to our collective work. They're really putting on the map the work we have done. Diversity is a real precondition for quality. We cannot have quality with like a subset of our trainees selected by any means. And so some of the things that have been learned in the field are very important is define your goal up front. What do you really want to accomplish? Is your idea truly innovative? Is it important? And is it doable? Is it feasible? Then, if yes, do you really need to launch a company? Can you license your IP to another company that already has capability to, you know, to do the production? I mean, if you just think about the recent example of Moderna, you know, when the vaccine was put together, it was not a new company being launched. It was handed over to the company that had like absolutely everything lined up to do the production. Something that I learned is incredibly important is to provide the proof of concept of the idea as soon as possible. Unlike academic research, you need to do your last experiment first. So if you're going to fail early before you spend your and other people's time and money. Build a good team. You need people that you like, that you share your passion, you share your interest, and you have this complementarity of the skills and expertise. So you can only succeed together. Get help and advice. It's just, this is what people don't do often enough. Resources are all around us. These are people, your colleagues, people who are going through the similar process, your tech ventures office, you know, like many resources that can really help you with the things that you don't know. So this is, I think, a sort of general. About promoting specifically the female scientists, I think we are doing much better than 20 years ago, because today is becoming rare to be the only woman in the room. If I use this as a readout, this has definitely changed. But there is so much more that we can and should do to support female scientists. What we all know is that the pipeline for professional careers in biomedical engineering in general and in tissue engineering is leaky every step of the way. I think it starts already in high school, you know, with these choices like, shall I study engineering or humanities? But the most obvious attrition I believe, is after graduate school, where many of our stellar students start to think that they need to choose between having a career and having a family. So this is where I think we need to intervene and to convince them that this is a false dilemma, that not only you can, but you absolutely need to pursue both. You will be a much better scientist if you have normal life, and vice versa. You'll be a happier person, you know, if you haven't sacrificed your career in order to have family. So for this to happen, I think we need to support strongly both individually and through like all organizations where we belong, this female talent. We need to encourage them at all career stages. We need to help them write the grants. We need to get them invited to give important talks, to be participating in important panels. They need visibility, and they will be opening doors for more. I mean, this is really how it works. And as I mentioned in the beginning, I think this applies equally importantly to other aspects of the diversity of our student and generally trainee population. You know, it's like besides sex, it is really the racial, social, and other backgrounds. I think the quality, passion for science should be really the criteria, and we should be blind to all other factors. But at the same time, promote those who are having like a little bit harder time to get aboard because of being disadvantaged in one way or another.
As someone who's first generation American and college student myself, I very much appreciate that message, and I'm sure those who are listening in our audience today do as well. And with great role models such as you, I think we're all inspired to keep pushing and keep going. And it's always a nice reminder to maintain a little bit of balance and not to just work constantly. But before we come to a close, maybe just a few rapid fire questions to cap things off. So I think the first for me, we've talked a lot about the professional today, and I would just like to ask something, talking about that balance again, personal quickly. How do you like to spend your free time? What do you like to do for fun?
I do lots of things. I mean, I love to read. I love movies. We love to invite people for dinners. I like to travel. I mean, I have family. Family is incredibly important for me. So it's really, and also, I mean, for me, the academic job is a lifestyle. So it's not 9 to 5, as we all know. I mean, it's like an integral part of what you do. So I mean, they are really like the different components just work together in a very nice way. And this is what I'm trying to encourage all my trainees to look into, because it is completely doable, and it's very important to really have this completeness.
Yeah, and to love what you do. Agreed. So as we come to a close then, do you have any other closing thoughts or shameless plugs, anything we might not have covered today?
Not sure about shameless plugs, but actually I would like to mention, to say that it's really my lab that is behind all of this. I have an awesome lab, and this is a large and very diverse team of students, postdocs, clinical fellows. What we share is really a passion for science and for its translation. As I mentioned, what we do is a lifestyle and not a job. We are at Columbia, and Columbia's mission is engineering for humanity. And this is really the mission that we embrace, because it means not only the focus on translation, but also focus on socially responsible research that can benefit everyone. Because I think this is something that has changed over time. I was trained that good technology is technology that brings profit. there is much more to that. And this much more is actually something that we are very interested in exploring. The other thing that I would like to mention is that my lab is also trying to provide service to tissue engineering community and communities. So we are a home of the National Tissue Engineering Research Center, where we develop tools and we develop technologies for tissue engineering that we make available. We also host a hugely popular seminar called Tissue Talks, where we have leaders in the field speaking for the worldwide audience. We have Science Simplified, where our students and postdocs have translated important science into simple language and bringing it to the general population and young students. So this is high school and even before high school. And we also have a Career Conversations podcast, where we are inviting some very inspiring people to talk about their career choices and actually to discuss with students that it's not binary choice as in the past, like you go to industry or academia. It's like a continuum of options. And some of my students want to be journal editors, the others go to law school. So this is this conversation. So this is actually where I would really like to close. I mean, it is a team effort. It is really the laboratory staying behind everything that I mentioned today.
I think that's the perfect message to close on. So thank you, Gordana, and to all the members in your lab for your time and for everything you do. Thank you again.
Thank you very much for this invitation.